Welcome dear students. In this video we are going to learn about a very interesting fact about the WNT signaling pathway. That is the role of WNT protein in the regeneration of a flatworm called Planaria. The scientific name of Planaria that is used in experiment is called Smithia mediterranea. This species or the flatworm Planaria can perform asexual reproduction by binary fissions. At first I will show you the structure of the Planaria, but I want to mention here that this is not the class of regenerations. The regeneration topic is under the developmental biology. I will make a separate video on regeneration, and in that video I will talk about more details about the regeneration of Planaria. But in this class I will show you only the portion that is the role of WNT protein in regeneration of Planaria. The Planaria is a flatworm. It looks like this. It has a head and the eye. This is the head and these are the eye. It has a pharynx and it has a tail. Very simple. I will not go into details, but it is actually look like this. It has a head and tail. Scientists, when experimentally cut this animal into two half, that means bisect. When they bisect these animals, the bisected animals, two region of the animals, it is the head and this is the tail region. They found that this upper segment of the fragment of the organism developed tail. It developed tail, and the lower segment actually developed the tail segment. The lower fragment of the animals developed the head. That is very interesting. So somehow this organism, the part of their segment or bisected part, know what has to become. Actually, Thomas Morgan, who actually the master of the fly lab, but he was actually in 1900, he was actually famous for his work on regeneration in Planaria. This is very interesting. He used to feed this Planaria by Drosophila, but in 1910 he starts working on Drosophila only, and he renunciated the work with regenerations. But he found a very interesting fact. He performed trisection of this Planaria. When he trisected this Planaria, he found that the upper segment, from this upper segment, a tail form. Let's suppose this is the first segment. From the first segment he found that the tail forms. From the third segment he found that the head forms, because this is the third segment, head forms. And also from the second fragment of the segment, this is the second fragment, he found that the upper portion developed head and the lower portion developed tail. That is very interesting, and he actually mentioned that maybe there is a molecule that helps in this regeneration, that is upper portion will develop, anterior portion will develop head and the posterior portion will develop tail. And that is the case of the WNT protein. WNT protein is actually a morphogen, acts as a morphogen. In my previous class I have mentioned that WNT protein actually acts as a paracrine factor. When it's secreted out of a cell, that cell produces the WNT protein, it do not actually diffuse very distantly. It remains very close to the cells because the extracellular matrix proteins express some proteoglycans that has the glycosaminoglycans like heparan sulfate or chondroitin sulfate. Example like glypican actually capture the WNT proteins from the source. So let's suppose these cells are secreting WNT protein. The cell that is close to this WNT protein become different cells, or actually develop different organ, or actually form a different tissue than those cells are present in distantly that has the concentration low amount of WNT. Here the concentration of WNT is low, but here the concentration of the WNT is high, and those molecules actually work like this. That means the concentration of the molecules, signaling molecules, have a very important role. We call that type of molecule called morphogen. Morphogen means those signaling molecules that determine the fate of a cell differently at different concentrations. If the cells is very close to the high concentration of the WNT, it will become different, or actually generate different type of genes, produce different type of proteins, and become different tissue. And the distant cells will become different or produce different proteins. That is called morphogen. It helps in the determination of different organs. So here some roles are present, and Morgan actually indicate this experiment. Trisections. He actually performed trisection, first, second, third. Okay. Now what happens and what is the reason behind the development of this anterior portion head and the posterior portion tail? Another thing I want to mention, when Morgan bisect or trisect this organism, and the segment, the middle segment is very narrow. Let's suppose here he has cut the organism, then from the first the development will occur normally, from the third development occur normally, but from the second segment that is very narrow inside, the development will not be even, because then the concentration gradient of this WNT will not be maintained. You have to understand this. If the segment is very narrow, then the WNT morphogen gradient will not be very actually active, then the structure will not form properly. Now I will show you the mechanism of this development, how it occurs. This is very interesting. They found that the middle segment, I am mentioning only the middle segment, any segment you can understand, but middle segment is very interesting. They found that in the middle segment, the posterior segment of this trisection, the posterior portion has increased WNT protein, high concentration of WNT protein in the posterior segment. Let's suppose this is the posterior segment. Here more WNT protein is present than the anterior segment. This is the posterior segment and this is the anterior segment. In the anterior segment, scientists have found a enzyme that is high in concentration, that is some WNT proteins can also be present here. Okay, some WNT proteins also is there, but the concentration of the WNT protein is very low in the anterior portions, and this enzyme is known as NOTAM. I have mentioned in my previous class what is the function of NOTAM. NOTAM is a hydrolase that is also a target of the WNT protein. If you have not watched my previous video on the WNT, you must watch the video at first. NOTAM is a enzyme that cleaves the fatty acid that is associated with the WNT protein. A WNT protein is actually associated with a fatty acid that could be palmitic acid or palmitoleic acid, and the addition of the fatty acid itself in the binding helps in the binding the WNT protein with its receptor called frizzled. If there is no fatty acid, this WNT protein cannot bind with the frizzled. And also the role of this fatty acid I have mentioned in my previous video, that if there is no fatty acid in the WNT protein, then the WNT protein cannot be secreted out by the cell. The WNT protein will remain in the endoplasmic reticulum. So this is very interesting fact, and which enzyme actually helps in this modification, that is called porcupine. I have mentioned in my previous class. I will not describe here the role of this enzyme. So NOTAM inhibits the WNT. That is the simple fact. In this upper segment or anterior segment, maybe there is WNT proteins are present, but the functionality of this WNT protein is low, because here the WNT protein do not have any fatty acid, and those WNT protein don't have the fatty acid is actually off instead. It cannot perform its function. So in a gist, I can write that where there is a high concentration of WNT, that will develop in the helps in the development of the tail, and where there is a low WNT protein is low, there will develop the head. That is the theory. Okay, now I will show you an experiment and the result is very interesting. Okay, when scientists take a control, they found that the development is normal. Anterior part head, posterior part is tail. This is the control. Every experiment must have control. It means there is no actually perturbations. Okay. Now they add a siRNA, that means that helps in the interference of RNA, that is called beta catenin siRNA. When scientists induced in the segment of the middle segment, when they introduce beta catenin siRNA. What is the role of beta catenin? You must know, because after when WNT binds with the result, beta catenin actually gets activated. That the beta catenin actually perform the main function. So beta catenin is very important. And if you inhibit this beta catenin by this siRNA, because siRNA interfere with the RNA of the beta catenin. This is one fact. It is called siRNA, small interfering RNA. So when this siRNA is introduced, the main fact is WNT will not work. So in this segment there is no WNT protein, and what happens? We know that if the WNT protein is not present, or the function of the WNT is not present, then it will develop head. And they found that the right thing, that in the anterior portions head will develop, and from the posterior portion tail should be developed. But in case of beta catenin siRNA, another head will develop. I want to mention here another time. We have shown here that the WNT protein helps in the development of tail and prevent the development of head. Prevent the development of head. You have to understand this. And another protein I have mentioned, that is called NOTAM. NOTAM, that is present in the anterior portion, prevent the WNT. So in the anterior portion head form, because there is no WNT. This is the circuit. This is the simple circuit. Now if I inhibit the beta catenin by siRNA, then what happens? This WNT pathway will not work in the posterior. And as we know that in the anterior there is a NOTAM, so NOTAM will downregulate the WNT. So from the anterior head will occur, but from the posterior tail should occur, because WNT promote tail. But as this pathway is inhibited by this beta catenin siRNA, so here from the tail will not occur, but head form. So bi-head means two head will occur, two head form. Now another experiment, they introduce NOTAM siRNA, NOTAM siRNA, so that NOTAM will not be produced. And they found a very interesting fact. They found that from the upper segment, what will happen? Do you have any idea? If we inhibit this NOTAM, if we inhibit this NOTAM, then NOTAM cannot inhibit the WNT, then WNT will prevent the head formations. So what will happen here in the anterior portions? Head will not form, because now WNT is active. It will become the foot or the tail of the animals. And also from the lower portion a tail will form. That is very simple, because there is no NOTAM, because we have totally inhibited the NOTAM by siRNA. So from the posterior tail will occur, from the anterior also tail occur, both tail occur. Now another experiment scientists have performed. They have tried to know what will happen if I add beta catenin siRNA with NOTAM siRNA. If I introduce both these siRNA, what will happen? They found another interesting fact, and I will conclude this class by telling the result. You have to understand also it, that what will happen, you have to think it. when we introduce beta catenin in siRNA, that means this WNT protein will not work. So will it form tail? Will it form tail? No, tail will not be formed because WNT protein will not work. If we block the WNT catenin pathway, WNT pathway by beta catenin siRNA, then tail will not form. So head should be formed. But we have found that if we use NOTAM siRNA, then tail also will not form, because NOTAM, when NOTAM is inhibited, then WNT actually form the tail. But in this experiment, we have also blocked the WNT pathway. You have to understand this. In this pathway, we have blocked the WNT pathway also. So WNT will not work. And the role of NOTAM is irrelevant, because NOTAM inhibit the WNT promote beta catenin, then tail form. If we delete this beta catenin, then tail will not form. And if we delete the NOTAM, then only the option is remain, that is head, because WNT pathway cannot inhibit the head, so head will form. And we see that from this experiment, both the segment form the head. This is very interesting. So this is all about the role of WNT protein in the regeneration of Planaria. Hope this video will help you. Thank you for watching, and always keep learning.